With Lecture 8, Presentation 3, we get to go to one of my favorite places in the whole wide world, the Fayum Depression of Egypt, to visit Oligocene primates. The earliest known relatives of platyrrhines, those are the New World monkeys, and catarrhines, Old World monkeys, apes, and humans, date to the Oligocene, 33.9 million years ago. The oldest fossils for both groups come from Africa and the Arabian Peninsula. So, the modern Haplorhini infraorders ignoring Tarsiiformes include Platyrrhini, New World monkeys, and Catarrhini, Old World monkeys, apes, and humans. Just a reminder, Platyrrhini traits include a dental formula of 2-1-3-3. They have lots of premolars, a zygomatic parietal contact, and no ear tubes. Catarrhini traits, in contrast, the dental formula is 2-1-2-3. We've lost some of our premolars. We have frontal sphenoid cranial bone contact, and we have a highly unusual ear tube. So, do the Parapithecoidea primates have platyrrhine traits? Well, let's take a look at where the red arrows are. First of all, by the teeth, the dental formula, 2-1-3-3. And back where the arrow is pointing to in the ear region, no ear tubes. Aha! The Parapithecoidea have platyrrhine traits. And do the Propliopithecoidea primates have catarrhine traits? Well, take a look at the arrows again. Dental formula, 2-1-2-3, that's a catarrhine trait. And they have frontal sphenoid cranial bone contact. So yes, these are catarrhines. The Parapithecoidea appears to be the ancestral group. The Parapithecoidea, with the dental formula 2-1-3-3, gave rise first to the Platyrrhini. Platyrrhine New World monkeys branched off of the Parapithecoids. Along with other African fauna, rodents and reptiles, for example, they probably rafted to tropical America. The Propliopithecoidea also diverged from Parapithecoids. These early catarrhines appear to have given rise to the Miocene Cercopithecoid Old World monkeys and hominoid apes. Did Parapithecoids behave like platyrrhines? And did Propliopithecoids behave like catarrhines? Parapithecoid adaptations: activity pattern, diurnal, small eyes for skull length. In locomotion, they were quadrupeds with even limb lengths. Diet: some were fulivores, they weighed over 500 grams and had crested molars, and some were frugivores with low rounded molar cusps. They appear to have behaved like medium-sized New World monkeys, such as the TD monkey couple up above. One thing to know about TD monkeys, just kind of interesting, these platyrrhines have mated pairings, and when a male and female are together in the trees, they twine their tails together. It's really cute. Now, what about the Propliopithecoids? Did they behave like catarrhines? In activity, they were diurnal, small eyes for skull length. In locomotion, they were quadrupeds with even limb lengths. In diet, some were fulivores over 500 grams with crested molars, and some were frugivores with low rounded molar cusps. They appear to have behaved an awful lot like macaque Old World monkeys. Macaques are one of the oldest of the modern Old World monkeys. This makes a lot of sense. And one Propliopithecoidea Egyptopithecus had sexually dimorphic traits. Canines. Male canines were larger than female canines. In total body mass, males were larger than females. In skull size, males were larger than females. And males had a male-only sagittal crest, as you can see right here. So this obviously is the male, and the other is the female. Otherwise, they have the same species traits. Sexual dimorphism Whoops, let's get rid of this. Sexual dimorphism among modern primates is most strongly expressed by modern catarrhines, the old world monkeys, apes, and humans. This appears to be a very old condition if we base it off of our Proplyopithecid ancestors. The best Parapithecoidea and Proplyopithecodea specimens come from the Fayoum in Egypt. The Fayoum is in the Sahara Desert, southwest of Cairo. Even though it is only about 60 miles from Cairo, it takes at least 3 hours to get there because of desert travel. So if you look at Egypt right here and you look at Cairo, it's right here. It would be way up here toward the top of this basin. This is an area where you get an awful lot of farming, etc. Then you follow the Nile down and you see all the beautiful green vegetation along the border of the Nile. And then you get to a large green area that is the Fayoum. Then there's a lake right here, the Birket Karun. And then you get the area where we find fossils. By the way, we cannot go into that lake. It's surrounded by quicksands and also some pretty nasty critters, so you don't want to go down into the lake itself. And then over on the right, you can see one of our field vehicles in the desert. In the late afternoon, particularly when it's really hot, the sands separate and become much softer, and it's much easier to sink your vehicle down into the sand and get it stuck. So you want to do most of your traveling in the coolest parts of the day. Parapithecoids are recovered in older rocks than Proplyopithecoids. So technically speaking, the first Parapithecoidea probably come out of this group here in the late Eocene. By the time we get into the boundary with the Eocene, we start having Proplyopithecoids right through here. And then after that, you've got both groups going off. Different localities require distinct recovery methods. For example, BQ2 is covered with modern sands, and the fossil level is a hardened butter-brickle conglomerate of ironstone and fossils. Dry sieving is used to find tiny teeth. So you get a shovel full of stuff out of the ground, you put it into your dry sieve, and you start shaking away. Doing this, my husband actually recovered one of the oldest known tooth combs. It was for a little galago. At L41, we have a huge quarry. Now, this place started out about the size of, say the top of a desk. Today, the L41 quarry is about half a football field in size. But if you notice at the back over here, see this high wall here of rock? That all had to be peeled off. Then you had to take off the layers down here to get down to the green layers, which is where you find the fossils. So it's a lot of work to skin it back and get to where you've got the fossils. At L41, a heavy top layer of hardened sediments has to be broken apart and removed. This exposes layers of distinctly colored, damp, and very salty clay stones that are almost waxy to the touch. It feels like you're touching melted chocolate bars, and the saltiness causes the skin on your hands to peel off. These are then pulled apart and chipped away to reveal the fossils, but you have to do so delicately. And I'll point out what's going on in this picture. You've got some folks carrying the rubble in these tire baskets. They're made from rubber tires, and they're going to shuck them off over the end of the wall over here. And then over to the right, you've got my husband, Tom. He is standing up here. I'm down on the ground digging in a pothole, and a friend of ours who joined the field season pretty late, Leanne Jolly, is digging in her own pothole for fossils. At L41, the fossils have lost their collagen, but they have not completely lithified. So you have to glue the bejeebers out of them and let them dry for a day or two before touching them. So Tom's holding Masrosector. It's a little faunivore in his hands there. That is after he's glued it and let the glue dry. Over on the left, we have Megalohyrax. This is one that I was recovering. You've got both sides of the lower jaw and all of the teeth. It's really a beautiful specimen. The problem is that I could glue the top part, but I couldn't get down into the bottom part. I had to let glue soak into it every day, getting a little closer to where the fossil actually is. I had to leave it in the field and cover it up for the year, and then the next field season the crew recovered it. Sometimes it's best to wrap a fossil while it's still in its matrix. So what kinds of things would we just wrap up? Well, I've got one I'm calling a scrapula here. Scapula is the name for a shoulder blade. This one was very scrappy. And then over here, this one here where I have the circle right up there, that's a frog. There's no way I'm going to get that frog out of that dry green mudstone. That needs to be taken back to a lab for processing under a microscope. It's fun, but you start getting foxhole fever working at L41. You dig deeper and deeper, and you keep popping up out of the ground like a prairie dog when a scorpion, or worse, shows up. What's worse than a scorpion? How about the things that eat scorpions? Salpugids. These little guys are scary. They do not see the world as we do. They sense it by heat and chemical receptors, and they don't judge size. All they judge is, does it have chemicals and does it have heat? Then I want to eat it. And they are fast, and they just chase people around in a circle. It's kind of fun when you're watching somebody being chased by a salpugid, but it's not fun if you're the one being chased. Some localities are covered with ironstone nodules that sit atop and next to the fossils. We collect these by surface prospecting, and it's hard on the knees and on the heel of your hand. And sandy sites require an unusual fossil extraction method. So this is a year when I wasn't out there. They've got camp pretty close to Quarry I here. Now it's much further away. But notice all these little piles of sand out here. Let me explain what's going on with all of that. We actually sweep the Sahara Desert. I know it sounds crazy when you tell people we get push brooms and we sweep the desert. The desert floor is swept in a great big circle, and the wind is allowed to harvest the next field season's fossils. We call this wind harvesting, and it was invented by this gentleman right back here, Dr. Elwyn Simons, who was in charge of Fayum research for over 30 years. So what you do is you sweep in a giant circle and you sweep back all of the sand, and then you leave for a year. And what happens is over that year with wind and rain, etc., you get the fossils popping up. It's the Brazil nut effect. If you open up a can of mixed nuts, you'll always notice that the biggest nuts, usually the Brazil nuts, are at the top of the can. All the little guys have worked their way down to the bottom of the can, and the large things are left on top. Well, when you do this sweeping job, what happens is the sand particles descend downward, and the fossils are too large to descend downward, and they're left up on top. So then the next year, you come along and you start scanning the surface of the area that was swept back the previous year. And you get lots of good fossils that way. So, by the Oligocene, primates with platyrrhine traits and catarrhine traits were present in the fossil record of Africa. And evidence from other fossil organisms and isotopic data suggests that the Oligocene Fayum had a warm and damp climate, supporting tropical habitat. Among the evidence, we have Fayum tropical plant fossils. This one down here is one of my favorites. It's an epiprimnon, and if you look, it's got all these tiny little cups. Those held fruit seeds, and then the whole thing was covered by a giant skin. It's a lot like a pomegranate, and you can find it in rainforests today. It's always found in tropical regions. And then up here, you can see in this butterbrickle, you've got another tropical plant. So you've got tropical plants. In some places, there are even tropical trees with giant prop roots. And we also have beautiful tropical animals. For instance, we have the bones of a wading bird, Eremopesis, and I know this is not the greatest picture. It was the best shot I could get, but right here you have the hip joint, here you have the back of the pelvis, and here you have the leg bone of a wading bird. Wading birds require water. And then over here by my lens cap, you can see the vertebrae of a giant crocodilian. So right through here. And then over here, if you take a look at this picture of a crocodile, you'll see at the top of the skull, it's got all these bones that are pocketed. These are dermal bones. They make up plates, and this is what gives the crocodile that pockety looking skin. Right here by my foot, you see a fragment of crocodilian skull. In one place, I pulled out a canine tooth of a crocodile. That was probably as big as my fist. It was just an absolutely huge crocodile. So we have tropical plants and tropical animals, and what this tells us is that the Sahara Desert of today was a tropical rainforest in the primate Oligocene, supporting little primates, as well as crocodilians, bats, manatees, some extinct forms of rhinoceros, early elephants, as well as hyraxes, tenrexes, possums, turtles, wading birds, you name it. It was a tropical zone. Now you are ready for Lecture 8, Presentation 4, where we finally meet the hominoids.